I'm going to start with something we all know, the plastic bag, a thing of progress. When plastic bags were introduced in the 1970s, they were seen as better than the paper bags that they replaced. They were disposable, they were cheaper, they were stronger, they used less material, and fewer trees are cut down to make plastic bags than paper bags. They're environmentally friendly. So much so that we used 1 trillion of them worldwide in 2001. Although we know that the disposable plastic bag is not really disposable, we continue to use things like this, the plastic water bottle. Plastic bottles are safer than glass, they're lighter so they use less carbon to transport them. In 2010, Americans drank 34.4 billion liters of bottled water, and that was 4% more than in 2010. This is better. Is this? Really? We solved the problem of convenient drinking water, but made a massive new problem of waste at the same time, and yet we still use them every day. The incandescent light bulb has been replaced by something better, the energy-saving light bulb. It uses less energy, lasts for longer, but while we threw the old ones out, these ones have to be recycled because they contain mercury, and many people don't recycle them. Recycling uses a lot of energy itself. Is this better? Well, it's complicated, but the plastic bag, the plastic bottle, and the energy-saving light bulb are all common examples of how design and technology can take a problem and solve it and make more problems in the process, all in the name of progress. We assume that design and technology makes things better. We take for granted that's what they do. But what do we mean by better? Cheaper, longer lasting, more sustainable, more high-tech. Better for whom? Whose better ultimately shapes our common future? We keep on doing the same things over and over again, and we expect different results. And there's a way that sometimes gets described, well, as this, insanity. So Einstein, he said a lot of clever things. This is one of them. Another one he said is this: we can't solve problems by using the same kind of thinking that we used when we created them. And I think, as a designer, that designers, we've got to get away from this idea of solving problems. And we've actually got to challenge those problems and ask different, more relevant questions about what we're making. So I work a lot with engineers, and engineers are focused on solving problems. But to me, design is more exciting. Design does something different. It's about possibility, life as it could be. It projects into the future. But designers call themselves problem solvers. And I think we've got to get back to this idea that design can help shape our future. So now I want to talk about how we might use design to actually ask better questions instead of doing the same old thing, solving the wrong problems. What I'm doing in my work is seeing, can we use design to actually affect the direction the technology takes before we even get to problem solving, to actually use design to alter the future? So I'm going to tell you about a field of engineering that I've been working in, which is very unfamiliar for me as a designer, and some of the projects that are trying to design different ways of thinking. And then I'm going to share some of the collaborations that I think hint at new ways of working. So, I'm a designer, but I became interested in this. This is a plate of agar jelly covered in E. coli bacteria that were engineered by undergraduates back in 2004 so that they go dark when they're exposed to light, like photographic film. And they called this E. coliroid because every biological design needs a good name. And this is a common thing in this field, synthetic biology. So this 2004 example is a really early example of this aim for biology to become more like an engineering or computer science discipline, that it could be standardizable, repeatable, predictable. Life, unfortunately, has some funny habits like living and dying and reproducing, so that's the challenge for synthetic biologists. But what they're trying to do is turn biology and life with it into a design discipline of sorts. And they believe that this will help solve the world's problems, that we could make the world better through materials and new kinds of foods and fuel and medicines. As an emerging field, I think it's a really interesting place to actually, fiddle about with what we mean by design. So that's what I've been doing, and trying to test out ideas, because at the moment, it's about this, problem solving. Let's make yeast make jet fuel, or get bacteria to secrete rubber to make tires, or the same kinds of non-biodegradable plastics that we already have. To me, this appears to be a disruptive technology, or a promised disruptive technology, that's actually promising to disrupt nothing at the same time. And I think we need to get away from this idea that we're just solving problems. How do we make yeast make jet fuel? Well, that's a big problem for the scientists. But we need to find ways to use less fuel and to question and invent alternatives that maybe don't use synthetic biology or use synthetic biology to find alternatives to fuel. And that's where I think we need to demand why we're using technology and what we want from it. And perhaps that's where novel and ethical innovation lies. So I'm going to share some of the projects now where we're experimenting with this, and I'll start with iGEM. So that Platobacteria was made by some members of this photograph, and this competition now looks like this. This was 2010. Now it's regional. I'm going to show you a bit of a video and tell you more about it. Echromi is an experimental collaboration between designers and scientists working in synthetic biology. In 2009, 7 Cambridge University undergraduates spent the summer learning the tools of synthetic biology, which is essentially a new approach to genetic engineering. Using standardized sequences of DNA in a format that's called biobricks, they learned to engineer bacteria. They designed their own biobricks using genes copied from existing organisms, inserted them into E. coli, and created bacteria that secrete colours visible to the naked eye. Echromi went on to win the International Genetically Engineered Machine competition at M.I.T. in 2009.
And joining us now is one of the winners of the iGEM competition. Welcome to Science Friday, Ms. Mullen.
Hi, thank you so much. I am part of the Cambridge 2009 iGEM team, and our project was called Echromi. And what we were trying to do is to improve bacterial biosensors. They're bacteria that can tell you the concentration of a pollutant in water. And they can do this because inside them they have a detector. So we developed 2 different parts, the sensitivity tuner, and this actually tells the detector when to turn on and when to turn off. So you have control over what level of the pollutant you're detecting.
And how does the bacteria show that it's on or off?
We use something called a color generator, which means that our bacteria changed color when the detector got switched on.
Wow, so they light up in a different color.
They actually change color visible to the naked eye.
So let's say if you put a swab of the bacteria in a polluted river, bacteria would just change color.
Yep, exactly. Though you'd probably want to put a sample of your water on a bacterial plate, maybe not the other way around.
Well, how would you envision something like this being used other ways in the future?
As designers, we worked with the team to explore Echromi's potential as they were developing it in the lab. And together we imagined a timeline proposing ways that living colour could evolve over the next century. These scenarios, some of which are shown in this film, explore the different agendas that could shape Echromi's use and in turn our everyday lives. One of the first real applications for this technology may arrive quite soon: a cheap disposable biosensor for testing groundwater contaminated by arsenic.
Bacteria could also be used to produce natural colourings and dyes. By 2015, there may be a profession of people who hunt for new pigments in the genes responsible, bringing them back for use in the food and textile industry. By 2039, you can go to the supermarket and buy this simple probiotic yogurt for cheap personalised disease monitoring. The yogurt drink contains Echromi bacteria, which establish a colony in your gut. They monitor for chemical signals that indicate the presence of a wide range of diseases. If they detect a disease, they start generating the corresponding coloured pigment, producing an easily visible output to prompt you to seek your doctor.
2049 sees the rise of the Orange Liberation Front, a terrorist organisation from the Netherlands, who are angry because a biotech company in China has patented the gene for the colour orange.
In 2069, Google releases pollution mapping bacteria into the atmosphere that turn red in the presence of excess CO2. And as the saying goes, red sky in the morning, Google health warning.
Our collaboration meant that Echromi was a technology that's designed from the start at both the genetic and the human scale, and with a long-term outlook. We found that design and science could have a meaningful exchange in the lab, which could prove useful when developing technologies in the future.
So I'm going to tell you about one part of this project. This is the scatterlog and its contents. And just very briefly, we actually mocked up these poos and we took them with us to MIT, to the iGEM competition, because we wanted to challenge something that we saw as strange in synthetic biology, which is this: biology represented as a machine, and to us, biology is a really interesting design interface. And if we don't want this, what do we want? And that was the question we were asking, to try and get the implications and the applications of a technology being designed at the same time. Because the impact of synthetic biology, if it comes to pass, will be scientific, economic, industrial, but it will also be ethical and ecological and personal. It changes the relationships between design and science, between nature and culture, and the things that we make and who we are. So this is the tree of life. We're down here with animals, and I began to wonder, this is a human-designed construct that we use to make sense of the living world around us. It's a design itself. So I began to wonder, where would we classify these new organisms that may have genes from all over the place, and perhaps we're going to need an extra branch of the tree of life to put them in. So I added one. This is the synthetic kingdom. It's just part of our new nature, an easy solution to an engineering problem. And what's been unusual is that it was taken up in a way. This is the front of the first textbook of synthetic biology. And it's a fiction. But it's a really useful fiction that enabled me to have amazing conversations with scientists who were willing to suspend their disbelief to actually talk about this classification issue. I remember being on the phone once to this Nobel Prize-winning scientist that was going on the front of a journal, and he was like, well, maybe you could move some of the labels, it's a bit inaccurate. And I'm like, what? This is unbelievable. And other scientists have said, maybe it needs to be smaller or bigger or more like spaghetti. And I don't know what the answer is, but whatever it is, it's a really useful fiction for getting us to think about how these organisms may not be so different from what already exists. Some say that by giving synthetic biology a kingdom of its own, I'm further separating this distance between products and nature. Or I think it puts our designs back into nature and actually removes this distinction between the things that we make and who we are. And if we make that connection better between nature and what we design, could we actually design better? So I'm going to finish up by telling you about this collaborative project that sort of really draws on all these ideas, synthetic aesthetics, which was funded by science agencies and enabled us to bring 6 artists and designers and 6 synthetic biologists together from around the world. They did residency in labs and in the design studios and artist studios, which is really unusual. And we got them to think about these questions. Can you design nature? If you can, if that's even possible, how would you design nature? And how might we design it well? So I'm going to tell you about 2 of the projects, just to finish up, which are very different. The first, we paired an architect with a plant scientist, and you'd think that there'd be nothing in common between what they do, but actually, Fernand uses 3D modeling tools to build models of his plant cells, and David uses genetic algorithms to design buildings. They became really interested in this. These are xylem cells from an artichoke. And it's amazing how as these cells grow, they actually compute these boundaries between themselves and their neighbors. And David and Fernand started working with a computer scientist to actually harness that logic and make it into a series of algorithms that could then be used to solve other spatial problems and design new structures. And what they're doing is actually challenging this core belief in synthetic biology that programming DNA is what makes a biological computer. And their project harnesses the logic of the whole cell and this natural logic and actually says, well, this could be a different model. And in doing that, they're opening up new space for different ways of thinking. And I'm going to finish up with this project where we paired a biologist and a smell artist. And they want to, what's a smell artist? I'm not even going to get into that. You have to Google Sissel. They wanted to challenge this cultural fear that they saw that we have of biology. There's this problem that we live in this antibacterial, fully pasteurized world, and yet we're being promised this amazing future that's powered by synthetic biology. So they wanted to address this. This is the human microbiome. So we are full of bacteria inside and out. 2 to 4% of your body weight is actually bacteria. And we're increasingly understanding how much we rely on them to actually exist. And some of those bacteria are elsewhere. Limburger, if there's anyone in the audience who's smelled Limburger, it smells like feet because it's the same bacteria on your feet as in Limburger cheese. So Sissel and Christina at Harvard Medical School in the lab started to wonder, could they make cheese out of human bacteria? Well, yes, this is how cheese is made. You stir the curds like this, and some definitely get in there, and this is not an unusual process, but this is a less usual process. So they took some samples, and I got to go to all the residencies and document the project. I was in the right place at the wrong time. Daisy's armpit cheese, best smelling. I wouldn't be showing it otherwise. So this is not synthetic biology by definition, but what's really important is that context is everything. Why do you think this is gross? This is actually, if you think about it, it's not that gross. We can debate that in the bar afterwards. But the thing, bacteria in context in our bodies or in our food means very different things to us. It's a cultural construct. And it's the same for the things that we design. We can't isolate them from the world that they operate in or from our behaviors, like the way we use a plastic bottle. It's not a disposable item. We are what we eat. But in a synthetic biology future, it could be that we eat what we are. And bacteria and organisms that we're engineering are closer to us than the abstract circuit diagrams and machine images can show us. Human cheese makes our symbiosis with biology and bacteria smellable and tangible. So I'll leave you with that. But we've written a book that's going to come out next year, which really kind of is this collaboration between so many different people that looks at these issues about designing nature. And it's key that it was funded by science and engineering. So the ideas are going straight back into the scientific community, as well as the policy and design and arts discussions. Implications are being brought into the engineering design process. And I think we really need more thinking like this that's experimental in this way. So to sum up, I've tried to say that we need to question the problems that we think that we're trying to solve, and given some examples of how I'm using design and art earlier in the development of a technology to try and sort of open up new areas of thinking. I think fundamentally, if we ask better questions, not only will we find better solutions, but we'll have better problems to solve. Thank you.